Hello and welcome to our panel. My name is Leigh Brody. So as Simon said, I'm new here. I'm an investment manager in the UCL Technology Fund, and I'll be chairing the event today. So we're going to be discussing safety in gene therapy. We have 4 fantastic panelists. So before we get into discussions, just a little housekeeping. I want this to be engaging and interactive. So I encourage everyone who's listening in to feel free to ask some questions as we go along. So there's a chat panel on your right. If you just kind of click it, open up some questions, we'll try to answer it as we go through. We'll do our best to kind of get through. So we have some fantastic panelists today here. I believe they'll join in a second. Okay, so we're going to keep going for a second. So I'll tee up what we're going to talk about today a little bit. We've talked a lot about gene therapy as a whole in the field. Obviously in the fund, we've looked at gene therapy. And while the field has made huge strides in technology, one thing that has placated a little bit is safety, of course. And that's a question that has come up. So we're going to talk today about some safety aspects that have come up in the media, and really get their opinion on how to address this point every day, from planning, indications to go after, through to manufacturing, and really the entire process. So without further ado, let's let them join us here. Hello. Okay, let's meet our panel. So I'll do a quick intro on each of you. Thank you for joining. I'll start at the bottom here. Professor Paul Gissen is a professor of pediatric metabolic disease at Great Ormond Street Institute of Child Health and a consultant at Great Ormond Street Hospital. I'll keep going to the bottom. Professor Amit Nathwani, professor of immunology. He focuses on the development of novel treatment avenues for hematological disorders and cancers using gene therapy and cell-based therapies. He's also the founder and scientific advisor of Freeline Therapeutics, and is currently the founder and CEO of Novelgen, which is one of the companies we just heard of earlier in our showcase. Going up, Gabriele Lignani. Sorry if I said that surname incorrectly. Gabriele is an ERUK Emerging Leader Fellow, an MRC New Investor in the Institute of Neurology, focusing on cutting-edge technologies such as CRISPR, activity-dependent promoters to develop novel gene therapy approaches for intractable neurological diseases. And then Claire Booth, gene therapist and pediatric immunologist from Great Ormond Street, leading the clinical gene therapy program and a PI in a lot of lentiviral CD34 positive trials of rare inherited diseases. Heavily involved in the GMP manufacturing, advanced therapies, and medical products as well. So first of all, thank you all so much for joining us today. So I gave a little spiel at the beginning of what we're going to talk about today, just to kind of go through that again. So we're here to talk about the the topic of safety in gene therapy. We're going to open the discussion up to broad points of the field, but we can kind of stick to that. And looking at the attendees in the audience, it's quite a technically savvy group. So we're going to skip over the basics of what is gene therapy and kind of get right into some questions. And so, when we started thinking about doing this panel and inviting everyone on and what we're going to be discussing, we thought about what's happening and maybe some current event stories going on as well. And one story that came up is where we'll start with questions today. So it's a bit of the elephant, I guess, so to speak. And the story I'm referring to is the Bluebird Bio story. So in February, a couple months ago, they hit pause on selling their Zentalgo, a beta thalassemia therapy in Europe, even though 2 of the cases of cancer, which happened in the drug sickle cell disease trial. So, of course, Bluebird launched an investigation afterwards. They came out stating that the lentiviral vector as a cause for this cancer was unlikely to have played a role in these events. But, it has given the field a little bit of a pause and has struck the conversation of, where is safety and how do we view it today? And does stories like this, how does it change the discussion at all? So I kind of wanted to open up by hearing your reaction to it. And if it does, how does it change your thinking about the use of gene therapy? Or maybe how do you read stories like this and interpret them towards your own work? Amit, I might start with you.
No, that's great. So clearly, any adverse event in any gene therapy trial has an impact on what we're trying to do. Most of us are trying to move forward with gene therapy programs that are at an early stage in development. And so we have to take note of any adverse events that come. But at the same time, we also have to be, good enough to be able to differentiate what we're trying to do for our group of patients from what is happening in the wider gene therapy community. And so most of our work involves the use of AAV vectors, which are administered systemically without any chemo conditioning. And so we feel that the toxicities that have been observed with the Bluebird Bio program don't apply to us in that setting. We have, of course, some of our own other problems, and these are problems that we have to confront and try and see if we can address and try and minimize risks of, say, liver inflammation, which is a problem with systemic administration of AAV, and look at ways of how we could do this, either by using a lower dose of the vector or by using immunosuppression to try and curb the severity of transaminitis that we see after systemic administration.
That's a good point. Claire, being on the other side of this and, being on the more maybe GMP side and running some of these trials, what are your thoughts on this?
Well, sure. When a story like this comes up, there's always going to be a lot of concern initially until further information is understood. And as Bluebird did, these things need to be fully investigated and sort of completely assessed. But I think it's also important to understand that some of these, there may be other factors at play other than the lentiviral kind of integration itself. So there may be disease-specific factors. So I think it's important not to necessarily tar every disease with the same brush, that there may be specific factors that are important. But obviously, there will be concern given the previous leukemogenesis and myelodysplasia with the gamma retroviral vectors. So there is the risk with the lentiviral vectors, but so far no causality has been shown.
Mm-hmm. Yeah, it's a good point, because I think always the question, at least, thinking at the wider scale is, do these kind of stories and the reportings, do they dissipate concerns over the use, and does it cause a ripple effect on the field as a whole?
Yeah, I mean, I think it's really difficult once this, although the story has to be reported, then the follow-up also has to be reported. So if there is no causality found, then that has to be reported. But of course, it's the first thing that people will remember is that, there was a problem. Mm-hmm. And not necessarily that actually there was no causality shown from that particular problem. So yeah, to a certain extent, I can see it does dissipate it, but I think we have to be sensible and step back and look at the investigation and look at the disease.
Yeah, it's a very good point. So maybe looking forward on that point, how will next generation viral gene delivery technologies improve gene therapy? And, is this still a main obstacle, a primary obstacle to go over? And, when you think about the next generation technologies, will it be aimed at making them more effective or safer? Let's see, maybe Gabriele, I'll ask you this one.
Yes, hi. First of all, I want to say that I'm working at the preclinical stage on developing these tools, and I'm working on gene therapy for brain diseases. So it's a bit different of for what we discussed before in the first questions. And I think that in my field for developing gene therapy for neurological diseases, I think that the next generation of viral delivery, viral tools will make a difference for sure, because one of the major challenge for targeting the brain is the delivery. And I think that the other most challenging things in treating the brain is also cell specificity, and will be also the challenge also to target all the brain, so have a wide expression. And I think that there is a big effort in the community now to developing new tools, new vectors to try to achieve this. So I really think that that in the next few years we can see an advance that will have a significant impact in the gene therapy for neurological diseases.
And maybe Amit, bit of feedback. I was going to say, Amit, in your role, how do you address kind of questions like this and thinking about where to go next?
I think, we have focused on trying to improve the potency of vectors. So we're essentially using lower doses. We've tried to improve the quality of the vectors that are put into patients so that eradicates impurities that might be responsible for the toxicities. And overall, what we have observed over the last 10, 15 years of working in the clinic is that the performance of at least the AAV system is getting better. We are becoming smarter about the capsids that we're taking forward into clinic. And in the majority of instances are able to reduce the doses that we are administering into patients for a given disorder. And so for hemophilia B, we've been able to reduce the dose level substantially whilst improving the level of expression that we see in patients. So, I think this is going to be the way forward. And as we learn more about how we can improve the quality of the preps that we're putting into patients, and how we develop smarter vector systems, the next generation of vectors, I think we will see fewer toxicities, providing that we use these vector systems with care. Of course, every drug known to humans is associated with toxicity. We've got to recognize that, but we have a responsibility to try and minimize the toxicity for our patients.
Mm-hmm. That's very true. Okay, I have a question just kind of on the broader scope, and Paul, maybe I'll ask you this one. Expanding the scope of where gene therapy will go. Do you think it will be used for common diseases or conditions at one point? Things like epilepsy, diabetes, heart disease.
Thanks, Leigh. Yes, I think so. There is no reason why it can't be. We need to understand the molecular basis of the common disease better. And once that's understood better, then for sure. So, for example, Gabriele is already working, targeting epilepsies. So when there is better understanding and when there is a common gene that you could, for example, do a gene addition and you can target, then maybe that would be helpful. So epilepsy is a good example. Some companies are already going for targeting diabetes. There is quite a lot known, but obviously there are different pathways there, and there are different types of diabetes, apart from just type 1 and type 2. So there are many others where gene addition is possible. Cardiovascular disease, there's already work being done. Cancer, for sure, where you could target cancer cells and kill them by using gene therapy. For infections like hepatitis, again, gene therapy can be used. So yes, there are a number of common diseases, from obesity, diabetes to epilepsy, meaning that you need to target different tissues. And I think you talked about where the advance is going to come, and we need to be in some tissues like liver, we need to target better and improve the targeting, and already new vectors available. For some tissues, the targeting is actually not great with the current vectors, for example, kidneys. And again, better targeting vectors need to be available, and targeting specific cells in the kidneys as well. So there is a lot there to be developed. But yes, I think there is also a lot of work being done.
Yeah, so naturally, I have to ask Gabriele on this, since working on this space and epilepsy. Obviously, you think the answer is yes. I mean, progress in the field, and how far are we from that?
Okay, I can continue a bit the question before, saying that actually myself with the colleagues, Stephanie George, Matthew Walker, and Dimitri Kuhlman, we are trying to tackle this important question in gene therapy field, to trying to find a way to have one tool, one gene therapy approaches to treat the common epilepsies, no matter what is the causes. So with epilepsy, we know that all independently on the causes, at the end you have seizures. So if we find a way with the gene therapy to modify the seizures and treat the seizure and stop the seizures, actually we can have a tool that can treat any patients without knowing even the cause. So I think that that can be a good example of how we can treat common diseases with that. And what was the other question? Was the second one?
Sorry, there was a few in there. I was kind of asking about how far maybe we are from it.
Actually, there is a clinical trial should start at the end of this year, beginning of next year. So we are not so far. Yeah. And in parallel, we are developing different tools to tackle the seizures in common epilepsies. So we have 3 or 4 different approaches that we are running parallel. So I think that in the next 5 years, we will know if it's actually is possible and if is a potential treatment for patients.
Great. So I'm going to squeeze in one question about manufacturing, because you can't really avoid gene therapy without manufacturing, even though this is safety. It's one manufacturing point. So you four are experts in the field. So I was wondering if you, in your imaginary world, if your respective CMC manufacturing departments or partners could develop one technology or improved aspect of gene therapy, what would it be? It can be anything from improving payload, purification methods, stable cell line, maybe an entirely closed system for clean rooms. Claire, might pick on you for this one.
Yeah, sure. So I think perhaps in the manufacture process itself, actually I think we've done pretty well with optimizing that over the years. We've got efficient vectors, we've got really efficient transduction protocols now. We can get high levels of transduction efficiency with really good viability. So actually, I think the products that we're able to produce are very good. I think the issues come before we start the production. So as I think everybody will say, the bottleneck of viral vector production, which the UK is addressing to some extent. But I think, as the field expands and becomes more crowded, actually, that may even get worse. Trying to find slots for vector production can be tricky, and of course cost. And I think that plays in slightly, if I might say, to this is going to be used for chronic conditions. Sure, it may be in the future, but if the cost is right. So for me, the issues around manufacture is really the reagents that you need for that, and that's true of editing as well.
Mm-hmm. Amit, I would love to get your take on this as well.
I think if there's one magical thing that we could have for AAV gene therapy, I would say that it would be lovely to have a stable producer cell line, partly because of what Claire mentioned, that that would allow us to reduce cost of goods ultimately, and that is important if we want gene therapy to be widely accessed across the world. Then we have to do everything we can to reduce cost of goods. And I think if we had a producer cell line, that may be possible.
Mm-hmm. And that probably ties in a bit to the kind of common diseases. That is one thing that would facilitate a common disease application quite a bit more. So what are some other maybe non-manufacturing challenges along those points? Maybe there are other struggles that other researchers, scientists, CEOs, CSOs are dealing with right now that you can kind of speak about. What keeps you up at night? Is it, access to the tissue of interest? Is it the therapeutic window for different diseases you're trying to treat, immune obstacles, payload, stages of diseasing? Maybe Paul, I'll ask you this one first.
Thanks, Leigh. So like Amit, we're working on using AAV-based gene therapy targeting the liver, particularly in pediatrics, though. And the issues that we encounter and have to overcome is the fact that the children grow, which is a great thing. But of course, as they grow, their liver grows quite dramatically. And the number of cells grow. And with AAV, as you target the cells, even if you target a lot of cells, as the liver grows, the cell dividing cells, they don't share the AAV. So there is a dilution of the effect as the child grows. And that's why as the time goes, potentially the effect of gene therapy can go down. So the kind of things that we want to overcome are how to target as many cells as possible, and then potentially redosing. And from that point of view, of course, that's difficult because there is a immune response to the vector. So that's something, again, we would like to overcome somehow. And there are various protocols and suggestions that groups are working on.
Oh, I think you hit mute by accident. There we go.
Have you lost me?
Just the
Sorry.
last few words.
Oh,
It's okay. We're all used to this now.
I can't hear you. Sorry.
Is it your headset?
I will come out and.
Okay. All right, he'll come back in. Amit, maybe you want to pick up this point if you don't mind.
Oh, there are many things that keep me
That could
awake
be
at
its own
night.
panel. Yeah.
But I think, we've had great success with AAV gene transfer in the monogenetic bleeding disorders that we've targeted. And that's been wonderful and fantastic. It's life-changing for our patients. But it is associated with toxicity, at least in the initial stage, which is essentially inflammation of the liver. And what we're trying to achieve at the moment is to try and find a way of controlling that, so that it doesn't happen. Because it's not a safety issue as such for patients, but it does lead to a drop in expression, and in some patients can lead to a complete loss of expression, leaving the patient immunized to AAV so they can't have it again, but actually no benefits from the gene transfer procedure. Mm-hmm. So we're working quite hard to try and see if we can control that. I think it's very important. But the one thing that we don't know, even after all these years of clinical success, is what is the biological life cycle of AAV in humans when we target the liver, as Paul and I are doing in our patients. And that's very important to know, because what do we tell our patients? We see from animal studies that AAV is maintained in an episomal format and can integrate, but actually the integration frequency is low. Yet we're seeing stable expression in humans for a period that now exceeds more than 10 years. And so how is that mediated? And we need to understand that and learn that, because then we can tell patients if they are theoretically cured or not. And I think that's very important. So we need to understand the life cycle. And so that's understanding the life cycle for efficacy reasons, but also it's important to try and understand life cycle for toxicity reasons. So we haven't seen any long-term toxicities in patients thus far, small number of patients. Mm-hmm. And we've been following them up for 10 years. But what's going to happen next, we don't know. And if you had an idea of where AAV sits in the cell, is it integrated into the genome? And if it is, are there hotspots? What are the implications? Then I think you can better guide and counsel patients. And at the moment, we're kind of limited in what we can tell patients around the safety and the long-term efficacy of gene transfer.
That's great. Paul, I just am checking you're back with us.
I'm back, and I hear that great. Amit finished what I was saying.
Oh, sorry.
No, that's great. Good to be back.
Yeah. So we've got a question from the audience. So I'm going to ask that now. And I think it's a good kind of point to ask this. So the question is, what do we think the longevity of expression will be? Who would like to volunteer to take this?
I can start. I think, the short answer is with AAV, we don't know. So far, it looks as though it's going to be a long period of time, potentially the lifetime of the patient, at least for hemophilia B, because we have the longest follow-up after successful gene transfer. And at the moment, we see a nice flat line when it comes to transgene expression of factor 9, which has remained stable now for over 10 years in some of our patients. So I think it will continue to remain stable. How long for? I don't want to mislead anyone, but I think a long time.
Great. So one more question from the audience while they're firing off. I'm going to ask them. Do we think gene therapy has been given unjust scrutiny from a safety efficacy perspective compared to a conventional modality? I'm going to ask for another volunteer on the audience question on this one.
You can go, Claire, if you want.
No, I mean, I think we covered some things, with relation to the bluebird issues. I don't think it's really been unjust scrutiny. I mean, these are experimental therapies, and we have a responsibility to put them under scrutiny and make sure that we are acting in the best interest of the patients and providing as efficacious a treatment as we can with a good safety profile. So, no, I don't think it's been unjust scrutiny really, but perhaps more scrutiny than conventional modalities like transplant, for example.
Mm-hmm. Yeah, Gabriele, did you want to add something?
No, I was only to say that obviously you are permanently change the genetic, the genome and the genetics of the cell of your cells. So I think that is justified to be cautious about safety and efficacy in this case. Compared, for example, for drug treatment, that you can stop the drug, for example, and it's a bit different.
Mm-hmm. Paul, do you want to add?
Yes, I agree with Claire that I don't think there is unjust scrutiny. The assessment of the this type of medicines for rare diseases, particularly, all of them go through the highly specialized technology committee at NICE, where I actually spend the whole day today. And any technologies like this, which are expensive and for rare conditions, will go through the same committee. When it gets to the common diseases, it will be interesting to see how that's going to be assessed. And of course, importantly, when Claire said that the cost needs to be different, of course, cost needs to be different, because of course the cost for these rare diseases, it's kind of accepted that if you're paying $1 million or more per shot, that's for a small number of patients a year, that can be acceptable. But if you're talking about hundreds of thousands of patients, that's not going to be acceptable. So it will have to come down very, very significantly.
I think that's true, but also really depends on what the cost of the current standard of care is. And if you look at hemophilia as a model, then a one-shot gene therapy that leads to long-term expression actually wins over factor replacement therapy in the hemophilias because it is so expensive. So we're spending over £100,000 per patient every year for the lifetime of that patient. And so the lifetime costs are huge. And that doesn't include, the hospital visits that these patients have to come for, because obviously factor replacement therapy is transient, and there are periods when patients are at risk of bleeding and have to seek medical attention. The advantage of gene therapy is that it is 24/7 protection for these patients, and the frequency of hospital visits obviously reduces. So overall cost of these new treatments may be lower if you take a long-term perspective on this. So the headlines may be that they may be the most expensive drugs known to humans, but it is a one shot, as opposed to, giving the therapy on a regular basis for a long period of time.
Well, absolutely. I actually completely agree that $1 million for a few patients, it's okay, because the hemophilia is still a rare disease. But if you're talking about something like obesity, which is, well, a lot of us are suffering from, if there is a gene therapy for that, then I think thousands will not be acceptable, I guess, unless, again, you take into calculation all the risks and over lifetime. Then maybe if the gene therapy will allow us to eat as much as possible, ice cream, anything, and stay thin, then perhaps that would actually justify the cost.
Except to the environment, because if we eat too much, then the environment will be affected. So I would like to discourage people from eating more than they need to.
I agree. And that might be another panel. How much are people willing to pay for this?
Yeah. I think just something else to put into that in terms of the chronic conditions and the cost and everything is, the risk benefit for those particular patients in the group, because if you're going to put something out that is going to be, applied to 100,000 people, the safety profile there has to be super tight. When you're treating a patient who's got literally no other treatment option, and they're going to die without this treatment, that's a very different risk benefit balance. I'm not saying we shouldn't make it safe. Yeah, we try our hardest to make it safe, but you see what I'm saying? Like it's a different balance. So like for obesity, to try and cure that with AAV.
You're right. You're right, and we already see that in the NICE assessment, when we are looking at treating patients with no therapies whatsoever, and the only thing we can do is palliation, then absolutely the risk benefit is different. Whereas in some conditions where, for example, dietary treatment is available and gene therapy comes in, then assessment is different. You're
Right.
absolutely right.
Levels of intervention, of course. It's interesting because I feel like Bluebird, so kind of circling back to the Zentalgo example from the safety, the CEO was constantly on this circuit of news, kind of battering back the price point because it was the second most expensive treatment for beta thalassemia. I think it was 1.8 million. And his first roadshow was about price. And then, his second was about safety. And so I think this drug kind of ticked both the boxes of arguments and conversation and debate that it's had to go through. Just a side point. Okay, so moving gears a little bit. And Paul, since you mentioned you were at NICE today, I think it would be good to ask one regulatory question, quite a general one, let's say. So my kind of question to you and everyone, of course, is the rate of technological innovation of gene therapy and cell therapy at that point, but we'll keep it to gene therapy, outpacing the ability of regulators? As a general point, are regulators too risk adverse or not enough?
It is a good question, but I'm not sure if necessarily the regulators are the ones that are slowing down the process. So I guess the process is quite long. For example, Amit now has amazing data in his hemophilia B patients, 10 years. However, is that drug? How close is the drug to being approved to go into clinic?
Oh, your headset, I think, again, Paul. We can't hear you. That's okay, because you've teed up Amit perfectly to answer this question for you while you re-enter the chat.
Can you hear me?
Oh, yeah. Okay, there we go. Yeah. Okay. Well, he'll re-enter. It's okay. A little difficulty. But Amit, you were kind of the next person I was going to go to naturally with this and he set this up for you. Thoughts about regulators. How are they helpful? Are they hindering on this point? Of course, very helpful to process, but are they keeping up with the pace of technology? And I can't guarantee there's nobody on this call from the HMRC, FDA, EMA, but...
HMRC I'm afraid of, but the MHRA I think we can take. Look, my view of the MHRA and the FDA is that they have been immensely supportive of these new advances. They are by nature cautious, but have worked with us to guide us on how best we can de-risk the programs that we're taking into clinic, quite often as first-in-human programs. And so a lot of the toxicities are unknown. We are all learning together, ourselves, investigators, patients, and regulators at the same time. And as we learn more, and as we improve the quality of vector and so on, then yes, I think the regulators are right in setting the bars higher for us to achieve if we want to take our products to clinic and ultimately to market, as Paul says, that we do need to get these drug products to market so they're widely available. And taking a drug into clinic as a phase 1 is a different risk proposition for regulators to getting a license to sell the product on the open market, where it is right that the bar should be set higher.
Yeah, completely agree. Paula, do you want to finish some of the bits you were saying?
Yeah, I don't know. I seem to be losing the losing the connection. Yes, I could see that it was kind of the idea finished. So I think there is a process that is quite long, and it's not really just the regulators that are there. So I don't think the regulators are the problem. I think the process is long, and perhaps, in fact, in the rare diseases, it's quicker than in common disease. So I think that people need to realize, the general public need to realize, and we often speak to the patients, and we say we're developing a new therapy, we're developing a new gene therapy, and I'm at pains often to explain that in the case of patients at the beginning, certainly in the kind of conditions I work with, the patients there are most likely not going to benefit because I work with some of the neurodegenerative conditions or progressive conditions. It's because it takes so long for the drug to get to the clinic.
And on a quick point to that, which also came from one of the audience members, is that do you think the COVID vaccine development has changed the risk appetite of regulators for approving modalities like gene therapy?
In some, so if I just start, in some conditions, they really desperate for gene therapies to come in. Like Amit mentioned that for in hemophilia, the clotting factors are so expensive. Similar in metabolic diseases, the enzyme replacement therapies, we easily pay 500,000 a year for life. So actually, the regulators are desperate for gene therapies to come in. If it's going to be one shot, even if it's $1 million, you can calculate the from that point of view, it's much better.
Yeah, very true. Claire, you were shaking your head there. Do you want to add anything?
Well, maybe I misunderstood. I mean, I think just in terms of the risk that is highlighted, just because it's a viral-based vector, I don't think it's the same or applicable at all.
Yeah. Okay, so we've got about 10 or so minutes left. So I just want to quickly touch upon one more modality that we haven't talked about today, and that's CRISPR. And expanding the topic maybe to things like base editing, prime editing, even epigenome editing that's been gaining a lot of attention, and how these technologies will survive the first few years and entering the clinic. Are these technologies to unlocking gene correction or things like safety? Is the risk profile maybe too high? Things like immune challenges around immune response, random integration, delivery issues, off-target issues. Are they too much? Claire, I might start with you on that one.
Sure. I mean, there's like 100 questions there.
I know. Sorry.
Areas to touch on. Okay, so I think one thing is some of these platforms are going to be useful for very specific niches. So, for example, base editing, that's not going to be applicable to very many monogenic inherited disorders where you've got different mutations scattered throughout the gene, and perhaps CRISPR editing may be more applicable for that. But I think so. On one hand, we've got highly efficient lentiviral vectors for some conditions, and that technology is moving forward, and that's great. And we can get those to the patients, and we potentially don't need any further gene editing for those kind of conditions. But there are going to be conditions where efficacy can be improved by using gene editing. I think we're still very much learning in terms of efficiency and safety. And it also depends kind of what you're trying to do. So for editing to knock out a molecule, so non-homologous end joining, that's pretty efficient now. The safety is, I guess, yet to be proven. But for the conditions that I work on, like monogenic inherited disorders, where you're relying on HDR, that's a trickier step, and the efficiency really kind of needs to be worked on. And then in terms of the safety and off-target and even sort of problematic on-target effects, I think we're still learning, and almost every week something will be published. Like this week, even with the sort of micronuclei story, something will come out that highlights another point that we didn't sort of potentially know about. So I think it's still a long road to sort of clinic for HDR-based gene editing. But these are really, really promising tools that hopefully once we understand a little bit more, we can work on optimizing the safety profile.
Gabriele, do you want to give your insights on this too?
Yeah, I'm working to develop a new CRISPR tool and gene editing tools, because I think that first of all, these new technologies are changing completely the game at different level, because instead of introducing an exogenous gene or a coding sequence, you can just modify the manipulated endogenous one. So it's changing completely the view, I think, of the gene therapy. And actually, I agree with Claire that depends on the pathology, but because obviously base editing or single mutation correction is not feasible at the moment, for sure, for diseases where mutation can be everywhere in the gene. There are other tools, for example, CRISPR activatory or CRISPR inhibitory, that actually you can, instead of reintroducing a new gene that sometimes the coding sequence is too big, cannot fit in AAV, you can directly modulate the promoter of that gene, the endogenous gene. And actually it's something that for me is very important, because the question that you asked before, what keeps you awake at night, is the dosage to me, because in the brain it's difficult to understand how much new gene you can add to not basically having another pathology due to the overexpression of that genes. So having a system that finally can manipulate and tune the expression of endogenous gene, I think that can be a good step forward. But I agree with Claire, there are still quite a few limitations, part of the immune response that in the brain is a bit different, but the delivery and the off targets, obviously. But I think that there is a huge scientific effort on the community, that probably one of the biggest in the history of the research, with a lot of groups working in parallel to trying to achieve and basically tackle all these hurdles of the gene editing. And as Claire said, every week there are at least 10 new research on this, on side effects, delivery, and long-term effect. So I think that is promising for the future.
Yeah, it's a good point. I mean, even Kathy Nikon's PNAS paper a couple of days ago, where she was citing that, approximately 16% of samples were large unintended mutations. And it kind of does beg the question, like, can you deep sequence yourself to safety on this? And, that's one way, right? But what are the other assays, and what is the combination of, maybe new gold standards we need for measuring things like off-target events or on-target at that? Okay, so we have a few minutes left, and I'm going to end it by asking the last question from the crowd, which is also the next question I was going to get to, so that's great. Do you think the field will ever move away from viral vectors towards novel vectors? So I was kind of thinking around, transposase systems or nanoparticles, things like that. Yeah, where do you think the field will go if they move away from viral delivery? Amit, I might ask you first on this one.
Sure, I think it's a great question. And my view is that the vectors of the future are going to look very different from the vectors that we're using now, because we're learning all the time, we're adapting and changing the vector systems effectively. The one thing that we do have to recognize is that viruses have evolved over time to be able to deliver their payloads efficiently. And we need to learn that in double quick time to be able to develop the next generation of vectors. But I think we will over time move away from vectors that look like viruses to synthetic vectors that are going to look very different.
Paul, any thoughts?
I agree that non-viral vectors would be very convenient, and for the problem that I was talking about before, and I hope you can still hear me. I'm a bit nervous about this. So for example, for growing liver and redosing, so a non-viral vector that doesn't elicit immune response, that would be ideal. For example, if you want to do in vivo gene editing, if gene editing can be safe, then redosing again, that would be potentially a big advantage. So I can see huge advantages of non-viral vectors, but they need to be there, of course.
Of course. Claire, it'd be good to get your point on this too before we go.
Yeah, I mean, I think we're doing pretty well actually in terms of the optimization of lentiviral vectors over the past decade. And yeah, I think there'll be some further kind of optimizations and promoter, but I think our field is moving quite a lot towards gene editing. So I think that for us will be the next sort of viral step.
Yep. And Gabriele, let's round it out. What do you think?
No, I think that will be great to move away from viral vector and having other vector, also nanoparticles that can deliver your payload. The things, for example, in the brain, the important things in the brain that you need to target precise areas, precise cells. So you need a way to target also your new vector to precise cell type. That is not easy. With the viral vector, you can do changing the promoter, but for example, nanoparticle is more complicated. Mm-hmm. So, but I think that's clear that I think that the field is moving on gene editing. I think that is the near future will be that, and then probably in the next 10 years, 20 years, probably there will be new vectors as well.
Yeah. Great. Well, this ends the panel session. I feel like we could have talked for hours, and I also feel like I got more ideas for 5 different panels I'm going to ask you all back soon for. So thank you all so much for joining me today. I hope the audience enjoyed it as much as I did, learned a lot, and yeah, quite excited about where the future goes. So thank you all, and I believe it's going to be handed over to the next session.
Thanks, Leigh.
Thank you.